The compound eye appears to be comprised of many hundreds or thousands of tiny facets. Each facet is actually the corneal lens for an individual photoreceptor unit called an ommatidium. Let us look at the detailed structure of an ommatidium. An ommatidium is divided into light-gathering and light-detecting components. The corneal lens is transparent cuticle that is secreted by two modified epidermal cells called corneogen cells. Corneogen cells secrete the corneal lens when new cuticle is formed at the time of the molt. The corneogen cells secrete the corneal lens, then later differentiate into the primary pigment cells. Below the corneal lens is the crystalline cone. This structure is either secreted or formed by four cells called Semper's cells. Light enters the corneal lens and is focused on the light-detecting apparatus by the crystalline cone. The light-detecting apparatus of each ommatidium contains 7 to 8 light-sensitive photoreceptor cells called Reticular cells. Reticular cells are actually photoreceptor neurons that detect wavelengths of light. The light-sensitive dendritic region of the reticular cell is the rhabdomere. Together, all of the individual rhabdomeres are referred to as the rhabdome. Rhabdomeres consist of parallel microvilli containing light-detecting visual pigment molecules embedded into their plasma membranes. Light entering the cornea and cone of an ommatidium is focused on and detected by visual pigments embedded in the rhabdomere membranes of the reticular cells. Reticular cells are surrounded by 12 to 18 secondary pigment cells so that each ommatidium can be functionally isolated from its neighbors. Ommatidia sit on a basement membrane in the retina, and axons from the reticular cells combine below the membrane to form the optic nerve that leads to the protocerebrum of the insect brain. Let us see how insect compound eyes function to detect light and perceive visual information. Let us now enter the compound eye and see the world as viewed by an insect. Here is how the world might appear to an insect from within a single ommatidium. Each ommatidium views the visual scene according to its position in the compound eye and the width of its lens. The view from each ommatidium is then integrated by the nervous system to provide a view of the entire visual scene.